Danielle, thanks for coming today. Very happy to have you.
Thank you for having me. Looking forward to it.
Yeah, we will get into the meat of the high subject that we have today planned soon. But let's just jump into your background and what you did maybe for schooling, and then some things you're working on today at the moment.
Yeah, so I got my PhD in biology from Temple University out in Philadelphia, Pennsylvania, and I started that program based off of some interests I gained during my undergrad at Penn State. So I volunteered in a deep sea biology lab.
Oh, nice.
And around this time, the 2010 Deepwater Horizon oil spill happened my senior year as I was finishing up. And I really got to be exposed to really the rapid response that my lab and other scientists kind of, conjured up and acted on to try to respond to the oil spill efforts in the marine environment in the Gulf of Mexico. So this really sparked my interest in deep sea biology, but also trying to understand how we kind of impact that kind of environment, which seems so distant and so far away.
Yeah.
So I started my PhD at Temple, trying to investigate some of those impacts on coral communities, and primarily focusing on deep sea corals because, in the first couple months after the oil spill happened, there were several deep sea coral communities found to be impacted, where they were actually able to fingerprint oil and some of the chemicals used to clean up the oil kind of like on the tissues of the corals that they found damaged and various stages of death. And so kind of focusing on deep sea corals to understand those impacts for my PhD, and using kind of genomic techniques, but primarily focusing on RNA sequencing. So trying to look at how gene expression changed as a result of those impacts, and how that kind of led to health decline and death. So looking at deep sea coral impacts through the oil spill was my primary focus of my PhD. And then I went on to do a postdoc at Florida International University, where I'm currently at again, doing some research, kind of a separate project, but looking at the evolution of bioluminescence in deep sea shrimp. So that's really where I got my first hand in trying to study bioluminescence, just using a different system, a different deep sea system. So this time focusing on shrimp or crustaceans, trying to see how that bioluminescence really influenced their visual capabilities. So kind of taking a different, focus with that kind of project, but using similar techniques. So using genomic techniques, high throughput sequencing of the bioluminescent light organs that the shrimp have that actually are emitting the light, but also of their eyes, again, to try to see how this light production influenced their vision, and their visual evolution.
Gotcha.
And then I moved on from there to do a postdoc at the Smithsonian, where I was, again, studying corals. I was in the Department of Invertebrate Zoology, working with the curator of corals there, and trying to look more at like how corals evolved through geological time, and also focusing on other deep sea communities. So looking at chemosynthetic mussel communities and how those kind of communities are connected, because we're still kind of discovering where they exist in the deep ocean, and trying to see kind of how they've adapted to survive in these really kind of extreme environments. So where a lot of sulfides and other types of toxic chemicals are being seeped from the sea floor. So trying to look at some adaptations that they have that allow them to survive in those communities. And then I kind of started this project looking at the evolution of bioluminescence in corals while I was doing this postdoc at the Smithsonian.
Okay. Yeah, that's awesome context. Let's jump into that, because that's the big news in my mind, at least. We were able to kind of pinpoint, or you and your team were able to kind of pinpoint more accurately when this evolutionary trait most likely first appeared in the ocean, correct?
Correct, yeah. So we were able to kind of use a lot of the kind of existing data that my team had produce for various other projects, kind of looking at the genetic similarities among anthozoan corals, and using those genetic similarities and kind of building those into phylogenetic trees to show those relationships. And then use kind of the presence absence of bioluminescence that we know of so far, including new records that we added with this study from opportunistic testing at sea, to then calculate kind of the probabilities along that tree, whether or not the common ancestor at every kind of point or node in that tree had a high probability of being bioluminescent. And we were able to predict with certainty that the common ancestor of all of octocorals, which is kind of a major class of anthozoan corals, was bioluminescent. So these are your soft corals, kind of like your sea fan type of corals, that are the kind of sister group to kind of hard calcifying corals. Yeah. So
these are the ones that are, I mean, when people think of coral, we think of kind of visible coral and the coral reef and all that stuff. These are the ones that are like way down, deep down.
So they actually are also in shallow waters. So if you snorkel or scuba dive, you'll see a lot of times like a purple sea fan. It almost looks like a purple tree.
I see.
It's kind of like has an arboreal shape. So there are shallow water lineages as well. So they're kind of exists kind of from depths all the way from the shallows to about 6,000 meters. But it is really these octocorals that do tend to get really deep.
Yeah, gotcha. Okay. Yeah. And why do you think, if you had to hypothesize or kind of estimate, that corals are the ones who might have had this first?
Yeah, that's a good question. So we just know that they're an ancient group of animals. So, if you think about the tree of life, we do know that there are a few older lineages, such as sponges and ctenophores, in terms of animals, metazoan animals. So in terms of animals that have a good fossil record, corals tend to be kind of the best. So the older lineages that we know of, including ctenophores, which do bioluminesce as well, they just do not have a fossil record. And that's because they don't really have hard calcifying parts that fossilize well, whereas all corals, including these soft sea fan corals, have some type of hard scleritic kind of component embedded in their tissues.
Got it. And when creating that tree where I guess you guys mapped out the different nodes and then the missing nodes, you could kind of fill in the gaps and kind of hypothesize where things fit. What went about starting from scratch and creating that? Did you have to start from scratch, or did you guys have data that you can work with to kind of get going?
Yeah, so we luckily didn't have to start from scratch because two of my co-authors have been generating these large data sets to try to help better resolve, actual species. And there's some misidentification and I guess, representation of what was actually a species or not in terms of corals, because they are so specious, they're very diverse. And there's also a lot of, on the opposite spectrum, there's a lot of like cryptic diversity, where a lot of things that like look very similar but are actually like very distantly related. So they've been working on this for a while and generating, kind of adding on to these data sets. So we were able to use kind of what they had and calculate these probabilities. We also added in a bunch of new data to see if we could kind of get similar support for some of the genetic relationships, and map the characters and the probabilities back onto that new kind of tree, that new data set, but the support was better excluding that new data, essentially. So we kind of included that with like the supplemental information. The story is the same. The common ancestor of octocorals is bioluminescent, and we're able to use fossils to predict the age of that.
Wow. And the age of that is in the realm of 500 to 540 million.
Yes.
Yeah.
Yes.
Got it.
So the previous record before that was in ostracods, which are a type of crustacean, which was 270 million years ago. So we kind of like effectively doubled the age.
Yeah. I mean, that's huge. That's huge news. I'm trying to think about what was swimming or walking around 540 million years ago, besides the coral.
Yeah, so this is the other exciting part of this is it was also the time of the Cambrian explosion, right? So like life on Earth was rapidly diversifying. We just started to get, animals evolving more complex like photoreceptors and eyes. And that's kind of the age of the predators kind of, emerged after this. So this idea that, at the same time that eyes and light detection was actually becoming efficient and useful for kind of capturing prey and survival. We also had this other form of light signaling that very likely impacted eye evolution and visual evolution. So I thought that was really cool and exciting.
Yeah. The eye grew and adapted in the ocean. So that's where it's been around the longest and had the most time to evolve, and evolution is really nothing without time. So very interesting to think about what came first. Was it the eye or was it the bioluminescence aspect? Because I understand that some corals don't necessarily use the bioluminescence to do defense or communication. It's kind of a byproduct in some cases of the algae that lives inside them. Is that correct?
Yeah, so some bioluminescence is produced by symbionts like algae, like zooxanthellae, but the ones in the deep sea are not using those symbionts. So they are creating all the components, for the reaction within their tissues, or they're obtaining some of it through their diet. But it is interesting to think about that, like what came first. This idea that bioluminescence, kind of its initial, I guess, function was not actually for light production, that was a secondary byproduct. And that, it was really to kind of eliminate free radicals, like having extra oxygen in the environment. So really trying to eliminate this excess oxygen in the environment, because the reaction consumes oxygen, and then light production is just a secondary byproduct.
Yeah.
So it's very possible that bioluminescence was around before, light detection was even happening in the oceans.
Right. And you could surmise maybe that if that's the case, life just realized, hey, this is so useful for many other things now that the eye is happening and coming and deep in the ocean. So let's talk about what some corals do use it as, in terms of is it communication, is it defense mechanism? And maybe I'd love to have you branch out into like other realms of more interesting realms of why bioluminescence exists in the deep ocean, at least, because it's on land too, but in the ocean.
Yeah, so for corals, it's a bit of a mystery.
Okay.
We aren't quite sure exactly why they use it, but there are a few kind of theories. So this idea that they are using it potentially to attract prey, because they will actually consume small, like crustaceans and zooplankton in their environment. And because a lot of other animals use it for that, it's very probable that they're retaining it for similar reasons. A lot of animals also use bioluminescence as a form of like burglar alarm to kind of deter predators. So it's also possible that, some coral species are using it in that way to kind of say like, okay, you bumped into the branch, of my colony. Now I'm going to light this up and kind of startle you so that you swim away and you don't, decide to take a bite, out of the colony. So it's very likely that it's being used for that as well.
Got it. So was the first one you mentioned but bringing in a larger predator, attracting a larger predator that might be able to eat the smaller predator that's attacking the coral?
So that's also another part of that, right? So if they are using it to kind of startle potential predators, it could be that either they're using the light itself to startle the potential predator, or they are again attracting a even larger predator that could then help them defend themselves from the would-be coral predator.
Gotcha. Yeah, you probably spent so much time on the how and the why, or the how in terms of like branching and tracking the evolutionary dispersion over time, but like sometimes interesting to just think about, like, why is this an evolutionary trait? 'Cause
Yeah.
I think in your study you said 100 specific cases that are independent from each other.
Yeah. Yeah, that's right. So right now it's estimated that there's at least 100 different cases that bioluminescence kind of convergently evolved across the tree of life.
In the ocean or across all trees?
Across the tree of life, yeah.
Okay, got it. Yeah, so that is compelling, obviously, in terms of how beneficial it can be. So, and then what other, so you mentioned shrimps. Let's dive into the world of shrimp and bioluminescence. How do they use it and why?
Yeah, so shrimp are pretty fascinating. So they have potentially two forms of bioluminescence. So some of the species that I have either have these light organs along their body that produce light.
Yeah.
And again, they make all the components for this reaction themselves. There's no symbionts in there. And they typically use that type of bioluminescence to kind of mimic light coming from the surface.
Okay.
So they emit it kind of as like a blue light to try to match the downwelling light so that it distorts their body profile in the water column.
I see. The sun is coming from the top, and they can shadow down, so they kind of balance it out with the light on the underside. Okay.
So visual predators from below would have a harder time kind of picking them off in the water column.
Yeah.
So that's one use for the shrimp. Some species have those, but have also evolved a secondary form of bioluminescence, which is in the form of this spew or vomit that they again create in their, internally. And when they are being attacked or startled, they'll actually emit this kind of bioluminescent liquid, and it kind of hides them. It's like a form of smoke screen. And then they typically kind of do this little like flip and kind of kick it towards with their tail, like whatever potential predator or danger is there, and then try to swim away before, the would-be predator notices where they are.
It's like an ink cloud for squid almost.
Exactly.
And it makes sense because obviously if you're at a certain depth, a fish's eye might not be, it's much less attuned to light than my eye. So that kind of light would probably just flashbang the fish, basically.
Exactly. Yeah. It's this really bright, brilliant blue color. It's very beautiful. And yeah, exactly. It kind of would take the fish like a little bit to regroup before it could actually like visually like hunt again.
Gotcha. So this is what arises, we can say, with 540 million years of evolution in the ocean with bioluminescence, is all these examples. What other examples exist besides the world of shrimp? And let's jump into like jellies and stay in the ocean though, but yeah.
Yeah, so a lot of, like I said, ctenophores and jellyfish, which are also cnidarians related to corals, are bioluminescent. And some of them have kind of accessory proteins that can make the bioluminescent color different. So a lot of jellyfish have GFP, green fluorescent protein. So they can actually kind of modify the color that's emitted of their bioluminescence using those, which is another cool adaptation of those. And a lot of fish have those photophores, very similar to the shrimp, but they have other adaptations. So they have like specialized photophores, some that are like near their eye that actually emit different colors than their body surface does. So like near red colors, and it actually helps them kind of recognize each other and hunt better. So a lot of different adaptations in the forms of these light organs or, some type of, bioluminescent kind of fluid or something like that.
Yeah. And let's talk about luciferin and how that comes into play and what that substrate is and why it's like, how prevalent is it, I guess, as well?
Yeah. So there's kind of the main reaction for bioluminescence is a luciferin substrate and then a luciferase enzyme.
Right.
And they kind of react together in the presence of oxygen. So that kind of enables the reaction. So this luciferin is, it's kind of used Luciferins, there's about, I think, 11 or 12 characterized luciferins to date. So they kind of overlap across the tree of life. So very distantly related animals can use the same luciferin. And a lot of marine organisms tend to use a particular type of luciferin called cilantrazine. This kind of cilantrazine luciferin helps kind of produce that blue color that's very common in the ocean. So the color that kind of is emitted by a bioluminescent reaction will really depend on the luciferin and luciferase combo. So again, you know, there's a lot of overlap across the tree of life, as in what luciferin's being used, but that luciferase is genetically inherited, and it's usually pretty species specific. So by kind of having overlap with the type of luciferin that you're using, even if you're not very closely related to another animal, you're very likely going to produce a very similar color of bioluminescent light.
And how do these species like enable or disable or heighten or decrease the amount of light, or is it something that they can't control and it's a different environmental aspect that controls it for them? Like there's a squid that I think does what you mentioned with bioluminescence on its underbelly to hide the shadow effect on the underneath it. But it doesn't, I don't think it controls it. I think it's just a matter of the intensity of the sun. So how often can these animals actually control this?
Yeah, so it kind of will depend. If it's a light organ, there's typically two kind of modes to control it. It's either kind of neuronal, so there's like you're perceiving the amount of light in your environment, and then you're adjusting the amount of light that your photophores are emitting. And then on other occasions, it could be hormonal. So there's like different controls, and in a lot of these systems, we don't fully understand which of these controls are actually, at play, or if both are at play. With things like the bioluminescent vomit, that's like, you can't control that, right? It's like, it's made in like your digestive gland, and you just like spit it out when you feel like you're being threatened. So that's always the same color, same intensity. And then it's just going to take you a while to regenerate all the components of the reaction before you can do it again.
That's so interesting. Okay. All right. So now let's try to talk about the eye a little bit more. You've done obviously a lot of work on the eye, or like adjacent work to the eyes in the ocean specifically. So like when I'm so fascinated because like the barrelhead fish, for example, like that is insane to me how that exists, and you can see the eyes and it just looks everywhere. So like, what work have you done on the eye, and how like what inspires you when you think about the eye in the ocean, and is there more to be found in the ocean relative to like evolution of the eye?
Oh, absolutely. I for the last point, I definitely think that there's a lot still to be investigated and understood. I think what kind of intrigues me, coming from the deep sea kind of background that I have, is that historically it was thought, because there's really just blue light coming down into deep waters past 200 meters, that most deep sea animals, below that zone can only see blue light. It doesn't make sense to see any other colors, essentially. So this idea that most visual capabilities of these animals are limited to blue light, has kind of been like widely, I guess, like hypothesized, but it's actually doesn't seem to be the case. It seems that there's kind of like a lot of variation in visual capabilities. And we see this in a lot of fish, and how their eyes have evolved these additional visual proteins to actually see what we think is a wide range of colors of light. And this tends to be linked to vertical migration throughout the water column. So a lot of marine animals don't just stay in the deep sea. And a lot of shallow water animals, including a lot of fish and shark species, actually migrate down to deep waters at night. So it's this idea that, the ocean is a lot more dynamic than we thought about. We think about land animals, right? And how they have ranges, right? And they have different kind of habitat ranges in terms of latitude and longitude. It's the same in the ocean, where a lot of these animals have pretty wide depth ranges. So they can actually be exposed to a lot of light variability. So for my investigations with the eye, even in shrimp, which are invertebrates and distantly related to these fish, is that these appear to actually have a wider visual capability than just blue light. They actually can see, UV shifted short wavelength light as well as really long wavelengths of light. So they can see beyond just blue, which is what a lot of my research right now is focused on. And I believe this is linked to kind of being able to distinguish between different bioluminescent animals in the ocean, right? You want to be able to recognize who you might be able to mate with or who you should run from.
Right.
Even if you're all producing some type of blue light, right? There's a range of color, even within blue, right? We just think of blue light, but it's a range. And then kind of adding on to that, I think it's because, again, they have this vertical range, and that environmental light has really influenced the evolution of their vision.
Gotcha. Okay. So since this is fresh, you're working on this now, we can stick on this for a little bit, like the shrimp and the colors of light they can see. So like us, we can see the whole spectrum almost. I mean, we can't see ultraviolet, or we can't see below what color? I forget exactly.
Yeah, so we could see short wavelengths of light. I forget exactly what that color is, but we can't see UV. Yeah.
We can't see UV. And you're saying they can see just blue and possibly green?
Well, I think that they can see blue, green, and I think they can also see like almost like a purple shifted color as well.
Okay. Got it.
So like not quite UV, but towards the UV end of the spectrum. So shorter wavelengths of light than we previously kind of thought.
And what wavelengths of light are other species and their same species emitting when they bioluminesce?
Yeah, so when they bioluminesce, they're all really bioluminescing this blue light, but it can kind of vary, and it could be species specific. And this might not seem like a big difference, but some, might be emitting wavelengths around 480 nanometers, whereas others might be emitting wavelengths around like 450. But by having different kind of, visual proteins that can see different wavelengths of light, they're actually able to kind of use those together to distinguish between what might seem like a very small change in that blue light.
Interesting. Okay, got it. And I'm having trouble thinking about, because I haven't searched it yet, but what these shrimp, because obviously I'm thinking about a different type of shrimp probably. So can you kind of outline to me like where they are bioluminescing? Because I'm trying to envision in my mind. I can't picture it exactly.
Like where on their body surface?
Yeah, like what are these the traditional shrimp that I'm thinking of, or are these more of the smaller zooplankton shrimp?
No, so I mean some of them are smaller, and a lot of the smaller zooplankton that you're thinking of are actually just juveniles of the bigger shrimp that you're probably thinking of, but they do look very similar to the type of shrimp that you would find and you would eat, in terms of, seafood.
Wow. Okay.
But in terms of their photophores, they tend to be kind of like right by their tiny little feet. So if you look kind of on their ventral surface, right where their appendages are kind of attached to the rest of their body, there's usually these like kind of brilliant red or purple dots, and those are the actual light organs.
Okay. And a photophore is what exactly?
Is a light organ. Sorry. Yeah. So a photophore is a type of light organ. Gotcha.
Interesting. And that can be with any organism that bioluminesces, whether it's a glowworm or a coral.
Yeah. So there's kind of like photophore is a very broad term for light organ, like one that's like on the actual body surface tends to be called a photophore, but they could be very structurally different. So some of them are very simple. Some of them have like lenses, so they can actually bend the light, which is really cool. And then
so they're not changing the intensity. They're actually, wow.
Some of them are, yeah. And some of them also have pigments that are actually changing the color as well, or masking the color. So it's very complicated. And there's still a lot to figure out, which is very exciting.
Yeah, that's amazing. So some photophores have lenses like a telescope or something where you can refract the light to change the intensity how it wants to?
Potentially, or the angle of the light as well.
Or the angle of the light. Wow.
Yeah.
Is that I forget the name. No, I don't. The blood belly. there's a squid that is, it might not be a squid, but it has the 8 rows going vertical where I think there's refraction or reflecting of the light where it's a rainbow up and it's like dotting up as it swims.
I'm not sure, but that sounds awesome.
Yeah, so there's something like that I was looking at the other day. And I think they refract or reflect as well. So that's just, yeah. So I mean, that's my point. There's nothing on land that does that, I think, and it just shows after all these millions of years how evolution can just do that in the ocean. That's pretty crazy.
Yeah.
So what's next for you in terms of are you focused on something to follow up the publish that you did in April, or are you doing kind of something else?
Yeah, so I'm working on multiple projects. So I'm still working on the shrimp stuff. I'm also working on another group of shrimp that have another type of light organ. So it's not just limited to those types of light organs, and it's actually located internally, but it emits a very like bright light that you can also detect outside of the body.
Yeah.
So I'm working on trying to understand the differences between that type of bioluminescence, but I'm still following up on some of this work with coral bioluminescence and trying to look at a couple different things. One, trying to understand. So, we talked about how we focused in this paper on octocorals, which are these kind of soft-bodied corals.
Mm-hmm.
But they have this sister group, the hard calcifying corals that also have some cases of bioluminescence, but they don't seem to have the same luciferase enzyme as the octocorals.
Okay.
So we believe that they potentially
branched off,
evolved like a different form of bioluminescence, so a different pathway. So kind of convergently within anthozoan cnidarians, you potentially have 2 independent origins of bioluminescence. So trying to understand that a little bit better. And also to kind of dig further within this octocoral group and looking at these luciferase enzymes and trying to see if we can develop a test just by using these sequences to kind of plug in new data to existing data to kind of say whether or not a species is bioluminescent or not based off of that luciferase sequence. So trying to make it less invasive to study bioluminescence and maybe a little bit easier in the ocean.
So you're mapping these from the different types of luciferase, and you can kind of tell that this is probably more evolved in a different branch because of how different this luciferase is, this version of luciferase, basically.
Yeah, in a way. So within octocorals, one of the other main findings of the paper was that there are 2 kind of groups within octocorals, and they differ a little bit by their kind of skeletal morphology and their kind of tissue composition. So we were able to actually show that bioluminescence is actually really only prevalent in one of these groups. But for some reason, both groups have retained luciferase gene sequences.
Okay.
So we kind of have this kind of preliminary tree of all of these sequences from corals from both of these groups. And we can kind of take a luciferase that, we sequence from a new species or from a species that hasn't been tested yet, plug it into that tree and see whether or not it's falling in with the group of bioluminescent corals or falling in with the group of non-bioluminescent corals. And then kind of dig even deeper, whether or not the lineages that have from our tree high probability of being bioluminescent, whether or not the luciferase is falling in with those corals or not.
Yeah, it stinks because we can only work with living organisms that are here today in this period in time on Earth. I mean, you can see fossils, but it's tough to you can't go back 2 million years, for example, and say, was it present here?
Exactly.
So, and are you using like any tools like CRISPR, Cas9 to do these things? I'm just curious. Or
not presently. No. So we're primarily just opportunistically documenting bioluminescence and trying to collect records and including negative records of bioluminescence just to get like a better summary of what animals are bioluminescent or not.
Mhm.
And we're taking small tissue samples and doing kind
Oh,
of shotgun high throughput sequencing and pulling out those luciferase genes.
got it. Are you getting the tissue samples, or do you have a team that does that?
Yeah, I mean, it's my collaborators have each kind of contributed tissue samples or observations of positive or negative records. So I've collected some, my collaborators have collected some, and the Smithsonian actually is a great reservoir of
Oh,
samples.
nice.
So that's been really good, trying to find ways to utilize museum specimens for this work as well.
Yeah, I'm sure they give you a lot of resources, so that's awesome.
Yeah.
Yeah, I'm just curious. Walk me through like some of the stuff you do in the day to day when you're like really working. Are you in like a lab? Are you writing a paper? Are you doing research from other people?
Yeah. So kind of all of the above. So a lot of days, especially when I'm, heavy in working on a data set and trying to kind of pull out gene sequences, I'm usually just in front of my computer
Yeah,
and kind of taking kind of turns, working on one project or the other, and then maybe writing up, portions of those projects or other papers. So that's kind of a large majority of my day to day. I also do, lab work or bench work where I'm actually, like going in and dissecting these shrimp animals,
Hmm.
removing those light organs and actually extracting RNA from either the eyes or the light organs. So very
Wow.
like detailed microscopic work,
Yes,
some molecular work there as well. And then on lucky days, I get to go into the field and actually collect the samples.
that's fun. Yeah, that's what I would look forward to, of course. But I'm sure you enjoy the computer work too. Are you using any software? I'm from the software world, so are you using any kind of specific tools or software?
So right now we primarily use a lot of bioinformatic tools to assemble
Makes
the
sense.
sequencing data and to try to like pull sequences from these transcriptome assemblies, because I'm using RNA for the most part. But for the corals, again, we were using genomic DNA,
Yep.
but using very similar bioinformatic tools to kind of assemble the sequencing data and pull genes of interest out. So a lot of bioinformatic tools go into it, and like doing gene alignments using software like that. I'm not sure if you're familiar with it.
No, I'm not, but it sounds interesting for sure. Um, let's switch things up a little bit because this is the topic that I like to talk about, or I want to learn a lot more about. But the world of like hydrothermal vents and cold seeps, and just it astounds me that, of course, you got like ocean snow. I know that's not the right term, but you know what I mean.
Marine snow,
Yeah,
yeah.
marine snow. Like that definitely creates ecosystems in itself, but there's this whole nother world that's actually just built from the ground up, from just molecules coming, or even like ions just coming out of hydrothermal vents from the mantle, I think, or the crust of the earth. And that's like not anything organic. And so people have surmised that's where life began. So how do you think about that whole world, and like how much research have you done there?
Yeah, hydrothermal vents are really exciting to me because that's actually the current project that I was hired for at
Mhm.
FIU, is to actually work on these hydrothermal vents, but our field work has gotten pushed a bunch since COVID.
Yeah.
So that is kind of like where my future is going to be, is studying these communities more,
Yep.
particularly looking at the crustaceans and shrimp that have, adapted to live there.
Yep.
And again, like you were saying, obtain like nutrients from, the sulfides and chemicals that are being seeped out from the seafloor.
Yep.
And trying to actually understand some of their visual adaptations to live in that kind of extreme, very hot environment.
Yep.
Previously at the Smithsonian, though, I did study chemosynthetic mussels. Bathymodiolus was the genus, and they have these really cool symbionts in their gills, which are chemoautotrophic bacteria that actually are able to take the compounds and sulfides that are being kind of extruded from these vents and cold seeps that are kind of other interesting kind of features on the seafloor that also seep a lot of toxic fluids and compounds out from the seafloor. And they're actually able to use that as nutrients, and they're actually able to then kind of transfer those nutrients to the mussels, and the mussels like survive that way just by getting fed by their bacterial symbionts on the seafloor. So I've looked at kind of how those mussel communities at these seeps and bents are kind of related and connected, since they do seem so kind of extreme and kind of separated by large kind of, I guess, areas. So their populations are very sporadic. So trying to understand how they kind of transfer larvae and how they transfer genes, from population to population. That was kind of what I was looking at. So they're really interesting communities. The animals that live there have to have these really unique adaptations to kind of survive. But once they have them, or they have a symbiont that can provide them with nutrients, they don't have to really compete for space because not many things can survive there.
Right. That's probably the benefit, even though it's hostile.
Yeah.
And so if either organism didn't have the other, I guess nothing would be possible down there. Like if we took away those bacterium right away, they would not survive, or they'd move away. Like, how does that work?
So the bacteria is also free living in the water column. So there are some that are symbionts with not just mussels, but also other species of like clam and other types of mollusks. Yeah. But they are free living, so they could survive technically without the mussels. But I did read kind of a recent study that I believe was done in Japan, where they actually transplanted like shallow water mussels into the deep sea, and they actually are capable of like taking up those symbionts fairly easily. So as long as they're around, even if they're in the water column, they can actually kind of filter the water through their gills and then obtain the symbionts that are in the water column near the seeps.
The same individuals that were in the freshwater?
So shallow water.
Sorry, obviously not freshwater. Shallow water, like.
So they were. I forget how long the study actually ran for. It was on the order of a couple of months though, and they were able to survive. It's pretty interesting. Yeah.
Okay. Wow. So it must. It's not an evolutionary trait. I guess it's just like it can happen instantly.
It seems like kind of opportunistic. I'm not sure what like that would lead to in terms of like overall fitness. So like how long would they be able to survive? Like, is it like a long-term or just a short-term like nutrient transfer?
That's true.
Would their like offspring actually survive or be viable? Because larvae, they might not be able to take up the symbionts as easily. So there's a lot to consider there, but it is interesting that they could just kind of survive and adapt.
Yeah, for sure. And the other thing, so I wanted to get back to something you mentioned at the beginning with Deepwater Horizon. I know you do, I don't know what you're doing nowadays with like building awareness for like mineral extraction and oil and all that stuff. But I guess let's start with like a little bit about like what your PhD was on and how you helped kind of bring awareness to that stuff.
Yeah, so I started my PhD again, like probably a year after the oil spill happened. And the lab that I was in was actually part of the rapid response that happened right around the time of the spill. So they were actually allocated the same type of oil that was spilled in the environment. So they were actually given like samples of that, as well as the chemicals that were used to try to clean up that oil, so that we could do some experiments to actually look at toxicity in like more of a kind of like an enclosed environment, like a tank, at sea. So some of my PhD work was actually looking physically at how the addition of oil and the emulsifier, which was called Corexit. It's a type of dispersant, kind of like a soap that they released into the environment for the first time in the deep sea during the Deepwater Horizon oil spill to try to prevent oil slicks at the surface. But unfortunately, the oil slicks still happen, and then they created kind of an inadvertent oil plume at depth that had a mixture of that marine snow that you were talking about, the
Oh,
oil that was being spilled, as well as the chemical dispersants. So it kind of created this kind of nasty byproduct. So I was able to actually look at how oil itself affected the corals, the corexit, this dispersant itself affected the corals, and how a combination of the oil and the dispersant affected the corals.
I see.
And from the experiments that I did, and they were kind of short term because it's hard to keep deep sea corals alive, at the surface, aboard a ship and happy. Kind of short term, 96-hour exposure studies were able to show that the corals actually did fine with the oil. I know that sounds, kind of crazy, like, the oil spill itself was the actual disaster, but oil seepage from the seafloor in small volumes is natural. So it kind of made sense that they were okay with the oil additions, even very high concentrations of oil additions, at least in the short term, they did okay. They did really bad, just kind of being exposed to that dispersant that was released into the environment as a cleanup effort. And then they also did really bad when that dispersant mixed with oil. So it caused all sorts of kind of physical damage to the coral colonies. And then I looked at that at, the gene level and looked at how their gene expression was kind of impacted to try to tease apart some of those impacts more.
Got it. Okay. And with the future of oil spills, because I assume we're going to continue moving oil, which makes sense. But what are the kind of new age technologies, if you're privy to that stuff, to kind of get rid of oil? I've heard microbes and things like that, and genetically engineering them to eat the oil. Is that like plausible or?
Yeah. So kind of in the aftermath of the oil spill, when my research and some of my collaborators' research on different, animals and different aspects of the marine environment, we're all showing the same thing, that these chemical dispersants were more toxic than the oil that was released, and that, it's not a viable cleanup option. A lot of other research was coming out, what could we do? A lot of experimental stuff instead of using this dispersant.
Right.
And I mean, some of the ideas were pretty wild, but it was using other types of like natural compounds, like cactus extract and things like that would degrade more easily in the environment, that actually worked in a similar fashion. But then other research came out that actually said that there were already oil degrading microbes that existed. Obviously, it makes sense. We have natural oil seepage from the seafloor. We would expect that things would have evolved to use that as like a carbon source, right? So they already exist in the environment. So
okay,
allowing kind of natural degradation to happen and trying to kind of mediate spills at the surface in like a different way, using more of like a physical kind of approach, is probably the best bet. But again, using more, like you said, more natural types of degraders, like microbes and other types of, I guess, like biotic types of dispersants, like using other types of plants or things like that, is probably the best way to go.
Got it. Yeah, because it's scary, because like there's just a lot of undiscovered things that we have yet to kind of realize in the ocean, in the deep ocean. So we don't even know what we're harming. We don't know what we don't know in that case. And I feel like when there's any type of ecology that's heavily studied, every day researchers go in and find something new. So there could be things that are the key to your research that are just about to go extinct, and then we just are that final straw that breaks the camel back. We don't know. So it's kind of it's a scary situation. I understand, we got to do our thing. We're capitalistic country. We have our needs. That makes sense. But any way to counteract that is important. So it's cool you did your PhD on that. And then just kind of a wild card question. With your upcoming research, what would be like something that you would love to have found out, or whether it's you or somebody else doing the research, or just a fact about something that would be like a missing puzzle piece in some of your research, that would be like kind of like a eureka moment to help us understand X, kind of bridge the gap between where we are and where we want to be?
It's a good question. And because I work on, very different things and have like various different research questions, it's hard to pinpoint one. But I will say, with kind of our future research with the hydrothermal vents coming up, we are also going to be trying to explore, document bioluminescence at these hydrothermal vents, which has never been done before.
Wow.
So really finding bioluminescence, some type of bioluminescent organism, documenting that at these vents, I think would be amazing. I'd be very excited about that. I think the hardest part about researching bioluminescence in the ocean is that there's still so many unknowns, and that was really a limiting factor in our study. Because anthozoan corals, again, which the study also talked about, we know that they are 771 million years old at least. So if we had less unknowns, maybe we would be able to date, bioluminescence back even further. So really just trying to document, more of the, positive records of bioluminescence, including in these really cool extreme environments like hydrothermal vents, would be pretty awesome.
Yeah, I feel like you're the perfect person to do that. Hydrothermal vents, bioluminescence. What are the odds that you or somebody else in your lifetime or my lifetime finds the evidence that it is actually that 700 figure that you mentioned with those other coral?
Yeah, I think if we're able to kind of unlock the mystery with hexacorals and try to understand what type of luciferase they're using, and really how that's kind of related across that other group of corals, I think that will really be the key to understanding whether or not it really is about 771 million years old. And I'm confident, hopefully not just in my lifetime, but hopefully in the semi-near future, that we'll be able to figure that out.
Wow. Okay. And do you think that's the cap? Would you be interested in other species, or is it like not likely at this point with the fossils we have? And
yeah, I don't think it's the cap. I think that'll just push the record. Like I said, there's this big debate that's been going on for a long time with scientists as to whether or not the most ancient animal is a sponge or a ctenophore. Wow. It's kind of funny, especially if you see some of kind of the heated arguments that some of these scientists get into. But we do know that ctenophores bioluminesce, and we know that they are the oldest animal, if not very closely the oldest animal on Earth. So it's very likely that bioluminescence is even older than that 771 million years old. But we just don't have that fossil record. So
sponge or ctenophore? Okay. Can you define animal in this context, how you're using it, just so we can be clear?
Yeah. So sponge, you mean sponge versus ctenophore, or?
Like, the oldest, because you're not saying the oldest living organism, you're saying the oldest animal.
Yes. So the oldest animal ever. So it's, whether it's extinct or extant. So the oldest phylum, I should say, of animal. Yeah.
Got it. Okay. Wow. So it seems to be the ctenophore, and those might hold the key to the longest.
But unfortunately, they don't fossilize well. Yeah. So unless there's some really amazing, discovery of a ctenophore fossil, who knows, right? The deep sea is amazing, or time travel. Yeah. Yeah. So until we have something like that could help us date ctenophores in general. Right now, I think corals are our best bet to at least have an understanding of the age.
Gotcha. And when can people look forward to that research you just mentioned?
Explore some hydrothermal vents along the mid-Atlantic ridge, so not too far off the coast of Portugal, and that'll be next summer. So it was supposed to be 2 summers ago, but it's going to be next summer. So it'll be, I guess, May 2025.
Wow. Okay.
Yeah.
That's super exciting. I think that's a great place to leave it. Danielle, thanks for coming on.
Yeah, thanks so much.
Excited for you to do that next May, and can't wait to look it up and look into it.
All right, great. Thank you so much for having me, Taylor.